All right, let's finish following our filtrate as it moves through the rest of the nephron. So we have two segments left in our story, and that is the distal tubule and the collecting duct. One thing that separates these segments from the prior segments, the proximal tubule and the loop of Henle, is that variable reabsorption occurs in the distal tubule and the collecting duct. In other words, I can adjust how much reabsorption of a particular substance I do in the distal tubule and the collecting duct. This is new for us. Up until this point, we really have only looked at autopilot reabsorption or obligatory reabsorption. In other words, it runs the same way every time and really doesn't get adjusted moment by moment based on the body's needs. Here, we are going to adjust the reabsorption and also a little bit of secretion based on the body's needs. Let's start by reviewing the distal tubule. So remember, in our nephron, filtrate goes from the proximal tubule, down the descending loop of Henle, up the ascending loop of Henle, and then to the distal tubule. As it makes its transition into that distal tubule, it crosses with the afferent and the efferent arteriole, and you can see that here, right there. So that is the juxtaglomerular apparatus. We'll explore the juxtaglomerular apparatus in more detail in subsequent lectures, but I just wanted to remind you that there is that talking that occurs between the afferent arteriole, the efferent arteriole, and the nephron as it becomes the distal tubule. So what does our filtrate look like as it's coming up into the distal tubule? Well, it has an osmolarity of about 100 milliosmoles, and it is mostly water, some sodium, some chloride, a small amount of potassium, and then some waste products. That's kind of the major gist of what is in our filtrate at that point. So then the subsequent question is, what occurs in the distal tubule? Like, what is going to happen in that tubule? Make sure you write this down. So the two major events that occur in the distal tubule are going to be sodium reabsorption and potassium secretion. And both of those are variable, so they can be adjusted. How much sodium you pick out of the filtrate or how much potassium you throw back into the filtrate. In addition, as you do sodium reabsorption, water is going to follow, so you'll have some water reabsorption there as well. Okay, the sodium reabsorption and the potassium secretion are linked. And what I mean by that is they go together and cannot be separated. They are under the control of the same hormone, and that hormone is aldosterone. So aldosterone is a hormone secreted by the adrenal glands. And remember, adrenal means adrenal on top of the renal gland. So the adrenal glands make and secrete a number of hormones, including aldosterone. So aldosterone is going to be the hormone that's going to end up controlling the mechanisms that I'll describe in a little bit. But when I say that they're linked, what I mean is that if I'm going to increase the sodium reabsorption, I also increase the potassium secretion. If I decrease the sodium reabsorption, I decrease the potassium secretion. I can't really separate them out. I can't say I want to increase the sodium reabsorption but not change the potassium secretion, unfortunately. So now let's go ahead and take a look at some drawings that are going to expand on the mechanism for how we can change the reabsorption of sodium and the secretion of potassium. So let's take the little boxed area that I have in pink and expand it. And you can see here I have a bunch of kidney cells. I'm outlining the filtrate, which has an osmolarity of 100 milliosmoles, the ICF, intracellular fluid, and the extracellular fluid, both of which have an osmolarity of 300-ish. I'm going to use a red dot for sodium and a purple dot for potassium. Now the first thing I'm going to do is I'm going to insert some sodium channels into the apical membrane of my kidney cells. These channel proteins are going to allow sodium to enter the cell and start the process of reabsorption. But my question is, how much sodium will be reabsorbed? In order to take a look at that, it may help if we just look at the gradients for sodium. Let's look at the same picture without potassium. So now we're just looking at sodium. The amount of sodium that comes in is dependent on the gradient. But the problem is that there's not that much sodium in the filtrate. So there's not a very strong gradient between the filtrate and the intracellular fluid. Without a strong gradient, not that much sodium is going to be compelled to cross that channel and go into the cell. So what establishes that gradient in the first place? Well, the gradient is established on the basolateral side through the sodium-potassium pump. So let's go ahead and add in some sodium-potassium pumps on the basolateral membrane. So that sodium-potassium pump is responsible for taking sodium that's inside the cell and kicking it out to the extracellular fluid. And you can see that I'm doing that in the left-hand cell here. By kicking out more sodium, I create a stronger gradient and therefore compel more sodium to enter the cell from the filtrate. So then it makes sense that the more sodium I kick out of the cell, the stronger the gradient will be. So how could I increase that gradient between the filtrate and the intracellular fluid? Well, one way that I could do it is by adding additional sodium-potassium pumps on the basolateral membrane. Let's consider that scenario. Here you can see that I'm more aggressively kicking sodium out of the cell, which draws more sodium in from the filtrate. And as soon as that sodium comes in from the filtrate, it's almost immediately grabbed by one of those many sodium-potassium pumps and kicked out. So let's summarize all of that. The more sodium-potassium pumps that I insert into that basolateral membrane, the more sodium I kick out, and therefore the less sodium is actually going to be in the cell. So there is less sodium in the cell, which means that I have a stronger gradient across the apical membrane between the filtrate and the intracellular fluid. The stronger gradient is going to compel more sodium to diffuse into the cell. And so that diffusion, of course, is facilitated diffusion, but the more sodium that comes into the cell, the more reabsorption I have. So what drives sodium reabsorption here? The number of sodium-potassium pumps. The more sodium-potassium pumps I have, the more sodium I'll be able to compel into the cell and therefore reabsorb. Now let's turn our attention to potassium secretion. In order to do potassium secretion, I need to provide a channel protein in the apical membrane. This will allow potassium to exit the cell and enter the filtrate, which is the final step of secretion. To increase secretion, I would need to increase the gradient between my cell and my filtrate, and the number of sodium-potassium pumps is going to dictate that as well. Let's consider scenario 1. I have a single sodium-potassium pump. I have a reasonably strong gradient that's going to push some of the potassium out of the cell through facilitated diffusion. Once I've reached diffusion equilibrium, no more potassium will be secreted. In order to secrete additional potassium, I need to reestablish the gradient by pumping more potassium inside the cell. But if I only have a single sodium-potassium pump, I'm only going to pump a little bit more potassium in the cell and therefore only secrete a little bit more potassium. So now that we've looked at that scenario, let's take a look at another scenario. With 3 sodium-potassium pumps in the basolateral membrane, I can pump 3 times as much potassium into the cell and thus create an increased concentration of potassium. That increased concentration of potassium will therefore also create a stronger gradient. By creating a stronger gradient, I can compel more potassium to leave the cell and go into the filtrate. So in this way, the number of sodium and potassium pumps in the basolateral membrane both dictate the amount of potassium secretion and the amount of sodium reabsorption. Let's add the effect on potassium secretion into our notes. Okay, so now let's go ahead and summarize some of this. Imagine a scenario where the body needs to conserve sodium. What I'm going to do is actually create kind of a logic train for you that you can follow along, and feel free to pause and write it out, that will kind of show you almost like a mathematical proof. If this, then this, then this, then this, and we finally get to our end. So I mentioned that aldosterone is going to control this, and we learned that the number of sodium-potassium pumps is certainly going to affect how sodium is reabsorbed and potassium is secreted. So you can intuit that aldosterone therefore controls the number of sodium and potassium pumps in the basolateral membrane of the distal tubule. Not anywhere else, just of the distal tubule. Okay, so let's look at this scenario. For some reason, my body needs to conserve sodium. Chloride is going to be implicit here. So I'm not using the word chloride, but chloride goes with sodium. So we're just going to assume chloride is part of the story as well. But we're going to focus on sodium. The body wants to conserve sodium. So if I want to conserve sodium, that means I want to keep as much of my existing sodium that's in my blood, which means that I'm going to want to reabsorb as much sodium as possible. In fact, we could reabsorb 100% of sodium. We rarely do that because in America, our diets tend to be very rich in salt, and therefore we're rarely in a massive sodium conservation mode where we would want to reabsorb every single bit of sodium. But all I'm saying is that it is possible to reabsorb 100% of the sodium if we needed to. But we're in sodium conservation mode. We're going to reabsorb as much sodium as possible. Okay, how do we do that? Well, the adrenal glands are going to secrete aldosterone. So the adrenal glands will secrete aldosterone, and then aldosterone is going to act on the basolateral membrane of the distal tubule cells. And it's going to tell those cells to insert more sodium-potassium pumps into the membrane. So again, just the basolateral phase. This, of course, is going to decrease the amount of sodium inside the tubule cells. So by adding more sodium-potassium pumps, we kick out more sodium back to the extracellular fluid, and the level of sodium in the cell drops. When the level of sodium in the cell drops, there is a stronger concentration gradient between the filtrate and the cell. And when there is a stronger concentration gradient between the filtrate and the cell, more sodium is going to diffuse in to the cell from the filtrate. And once we get it into the cell, one of those many sodium-potassium pumps are going to immediately grab it and kick it out. And so this ends up increasing the amount of sodium reabsorption. So this summary here shows you in kind of a mathematical proof way how the body's needs triggers the release of aldosterone. And then from there, we get a series of events that ends up increasing our reabsorption of sodium. Now, we can also look at the converse. So I can think of no saltier food, I'm sure it's out there, but this is what I think of, than beef jerky. If you eat a big bag of beef jerky and you urinate, an hour later, you will definitely smell the pungency that is reflective of all of that extra salt. So tons of salt, not really tons, but lots and lots of salt in beef jerky. Okay. So if I eat too much beef jerky and I eat all that salt, then all that salt is going to get reabsorbed into my blood. So now my blood has too much sodium and I want to get rid of some of that extra sodium. So what do? My adrenal glands are going to stop secreting aldosterone. When the adrenal glands stop secreting aldosterone, what this does is actually triggers the removal of sodium-potassium pumps out of the basolateral membrane. So again, absence of aldosterone or less aldosterone circulating in the blood equals take the sodium-potassium pumps out of that basolateral membrane. Well, when I do this, I'm not kicking out as much sodium out of the cells, so the sodium concentration elevates. My concentration gradient between the filtrate and the cell is not as strong as it was, and as it weakens, less sodium is going to be compelled to diffuse into the cell from the filtrate, and therefore I have decreased sodium reabsorption. Now I'm using sodium levels in my blood as kind of a trigger for which pathway I'm going to take, but know that potassium can also trigger one of these pathways. Specifically, if you have elevated potassium levels in your blood, you will trigger an aldosterone because you can't have too much potassium in your extracellular fluid. If you do, it ruins your gradients and you don't get the electrical changes that you expect in your heart, and therefore your heart will start undergoing heart palpitations because of electrical deficiencies. So your body definitely recognizes that an elevated potassium level in the blood is really bad and needs to be dealt with immediately, and so that will trigger aldosterone to increase potassium secretion so that you get rid of that potassium and let it go down the drain. Okay. Now that we have looked at the distal tubule, let's take a look at the collecting duct, which is quite a bit easier. So in the collecting duct, the major process is going to be variable water reabsorption. And because it is variable, it is also under the control of a hormone, and that hormone goes by two different names. If you are in my class, I expect you to know both of these names. I use them interchangeably. Antidiuretic hormone is probably the more informative of the names, but vasopressin is another name for the same hormone. So they are the same hormone. They just go by two different names. Why? I don't know. Okay, once you've written that down, let's talk about how we could vary the amount of water that we reabsorb in the collecting duct. In this image here, you might notice that the collecting duct passes from the cortex through the medulla. Now, why is that significant? It's significant because the medulla is going to be saltier than the cortex. In other words, the collecting duct is exposed to the same gradient that the loop of Henle is exposed to. The osmolarity of the extracellular fluid is going to be anywhere from 300 up here to 1200 down there. All right, this is kind of a busy picture, but bear with me as I show it to you. Ignore the blood for my purposes. What I wanted to show you was that we have filtrate. It's coming down the collecting duct. You can see the osmolarity increases. And so we have the potential to draw out a lot of water because we're being exposed to an increasing concentration gradient. If this is not clear why we would draw out a lot of water, then please refer back to the loop of Henle reabsorption lecture where it's shown in more detail. But by pulling that filtrate through an increasing salt gradient, water is compelled to go to where there's more solutes, which is going to be out here. If we allow water to exit. So remember, we can also change how permeable a membrane is to water. And we have two ways of doing that. The amount of cholesterol will impact the permeability of a membrane to water, but the other thing that impacts the permeability of a membrane to water is the amount of aquaporins. And for the collecting duct, the only one that matters is going to be the aquaporins. So it is the number of aquaporins that will dictate how much water is actually reabsorbed. In the image here on the left, we imagine that we have lots of aquaporins, and so it's very easy for water to leave. So in the image on the left, we have lots of water reabsorption. In the image on the right, we imagine that we take out all the aquaporins. When we take out all of the aquaporins, it is more difficult for water to leave, and so the water tends to stay in the collecting duct. Let's consider an analogy that will help us understand this. Imagine that I'm going to use PVC pipe in place of a collecting duct. On the left, I have continuous PVC pipe. There are no holes. But on the right, I'm going to drill a bunch of holes into that PVC pipe, and those are going to represent aquaporins. So when fluid starts going down my left duct with no holes, it just goes straight through to the bottom. There is no reabsorption, and all of it is collected as urine. So without aquaporins, I can't do reabsorption. But now let's consider the tube on the right, the one with all the holes in it. If I start pouring water down that, water is going to slip through the holes. In other words, some of the water is going to exit through the holes, and the amount of water that is left at the very end is going to be much less. So I'm going to collect a lot less urine in my bucket because I've done a lot more reabsorption. In the same way, by adding a lot of aquaporins into the collecting duct, you increase your reabsorption and decrease the urinary output, or the amount of urine at the very end. Again, adding a lot of aquaporins into the collecting duct is going to increase the water reabsorption. And consequently, the amount of urine that you make at the end, or how much is left over, is less. So we can measure the amount of urine that is produced. It's called urinary output. And we could actually measure the volume of it, right? So I could ask you to take little urine cups and collect your urine throughout the day, and you could measure how much urine you make. When you're really dehydrated, you don't make very much urine, because your body goes into water conservation mode, must keep water because you're dehydrated. And so it would make sense then that you would insert a lot of aquaporins along the wall of the collecting duct, making it very easy for water to exit, be reabsorbed, and therefore there's very little water left over. But what happens if you drink a lot of water? Let's say that I gave you a liter of water and you were drinking all that water really quickly. What happens? Do you have to use the bathroom in 45 minutes or so? Maybe 30 minutes? And when you do, what does that urine look like? Well, first of all, there's quite a lot of it, and it tends to be very dilute. Why? Because you overfilled your body reservoir of water when you drank an extra liter. So your body goes up, too much water, let it go down the drain, and so you stop reabsorbing so much water. And the way that you stop reabsorbing water, again, this is only in the collecting duct, doesn't affect loop of Henle or proximal tubule or even the distal tubule. The way that you adjust that is by taking out the aquaporins. If you take out the aquaporins, water just goes down to the end and is captured in our bucket or our bladder, and then it becomes urine. Okay. So I mentioned that antidiuretic hormone, or vasopressin, is going to control your water reabsorption in the collecting duct. Try not to memorize this. Try to reason it out. What does antidiuretic hormone mean? If I say the word antidiuretic hormone, imagine that I'm secreting antidiuretic hormone. What is that going to do? Well, what does that mean? Anti means against, and diuresis means to urinate. So against urine. In other words, no pee. So antidiuretic hormone is going to equate to less urine. So when antidiuretic hormone levels are high, you make less urine. So what does that mean? Well, if you have less urinary volume at the end, what that means is that in your collecting duct, you reabsorbed more of the water. Well, how do you reabsorb more water in the collecting duct? You insert more aquaporins into the collecting duct. So we can intuit then that antidiuretic hormone specifically causes the insertion of aquaporins into the collecting duct. So if my antidiuretic hormone levels are high or vasopressin levels are high, I'm going to insert aquaporins into my collecting duct and therefore reabsorb more water and produce less urine. What about the converse? In the converse, we have low levels of antidiuretic hormone, and then the collecting duct is going to take out all of those aquaporins, and the water is just going to go down to the bladder. You're going to have less water reabsorption and more urinary volume. Okay, this table quickly summarizes the major functions of the distal tubule and the collecting duct. You can go ahead and pause the video, read it, make sure it makes sense. The only thing you're not responsible for my class is going to be hydrogen ion secretion, which is not something I cover. We have finished our story of looking at the processes that occur in the nephron. So we've looked at filtration, and now we've looked at reabsorption and even a little bit of secretion. In our next lecture, we're going to learn about something called the renin-angiotensin-aldosterone system. I'll see you then.